In today's lesson, we'll be discussing the structure and function of macromolecules. So our goals and objectives are to understand the synthesis of macromolecules, to explain dehydration and hydrolysis reactions, to discuss carbohydrates, and there are three types: monosaccharides, disaccharides, and polysaccharides, to describe the four major types of lipids, and to differentiate between saturated and unsaturated fatty acids. There are four classes of macromolecules. We have carbohydrates, proteins, lipids, and nucleic acids. And these four classes of macromolecules are needed for us to survive. They're needed to support the functions of life. Now, the word macro means large. And if you remember micro means small. So our macromolecules are just two or more micromolecules. So these large macromolecules may consist of thousands of covalently bonded atoms with a mass greater than 100 daltons. And you should remember from the chemistry lecture that daltons are equivalent to grams. Now, when these covalent bonds are broken, they can actually enter into the mitochondria, which is where they're used to form ATP. And we'll talk about this later on in your cell lecture, but ATP is pretty much an energy currency that helps the cell do everything that it needs to. Everything that we do needs ATP. So a bond is formed during what's called dehydration synthesis. It's also known as condensation. So you'll take two simple sugars and you'll combine them into one larger sugar through dehydration synthesis. So let's break those words down. Dehydration just means to remove water, and synthesis means to make. So we are making this bond by the removal of water. So you'll see the short polymer and the unlinked polymer. And on a short polymer, we have a hydrogen at the end. And our unlinked polymer, we have an OH. So these two molecules will come together and leave the reaction as water, leaving us with a longer polymer. So we break bonds through hydrolysis, and this is just adding water to break down the covalent bonds between these polymers. So we can break the word hydrolysis down, and we know that hydro means water, and lysis means to pop. So we are just using water to break this bond to get the release of energy. So can you think of anywhere in the human body that or any animals would use this process? So when you think about digestion, that's the breaking down of food to macromolecules so they can be further broken down into energy for our cells to use. So carbohydrates are basically just sugars. A few examples would be bread or pasta or pretty much anything that's not a protein or a fat. So carbohydrates are used as fuel or as energy. They're also great building materials. So carbohydrates are made of carbon, hydrogen, and oxygen. And you'll also see these same molecules in lipids, but the amount is much smaller in carbohydrates. You have three different types. You have monosaccharides, and mono means one. You have disaccharides, and di means two. And then you have polysaccharides, and poly means many. So monosaccharides are simple or reducing sugars. They have 5 or 6 carbons, and they have a 1-to-1 ratio. So this N right here can be any number. You can plug any number in. If you wanted to do an example of 1, it'd be C1H2O1. So some common monosaccharides are glucose and fructose, which are found in OJ or orange juice. And these are cheap sugars that your body doesn't have to actually do any work to break them down. So we can use the example of orange juice. If you were to drink a glass, within 20 minutes, your body would be using that fuel. These are common, inexpensive sweeteners. There's no energy that's needed to break them down. So these need to be consumed in moderation. So monosaccharides can exist in either a linear form, which you should remember from the chemistry lecture that linear means straight, and here's our linear form of a monosaccharide. They can also exist in ring form. Another thing to remember is that in the ring form, it's harder to break down than the linear. Also, if you see a ring, know that any spot that doesn't have a molecule listed on it, like these right here, unless it's stated otherwise, these are carbons. Now we see right here we have an oxygen right here, so we know that that's an oxygen rather than a carbon. So disaccharides are just two monosaccharides that are joined together by glycosidic linkages. And that's important to know. So just note that if you have disaccharides or polysaccharides, you're going to see a glycosidic linkage, and you can see those right down here. It just kind of looks like the sugars are holding hands. So we talked about the two different reactions. So right now I want you to think about what a disaccharide, would that be a dehydration or hydrolysis reaction? And you see the answer right here, but you should know that's a dehydration reaction. If you remember from the previous slide, we talked about whenever we are making disaccharides or polysaccharides, that we have the dehydration synthesis. And you can see that here again, we have our hydrogen and our OH coming together, and we see water leaving from this reaction. Some other things you want to make note of when you have the monosaccharide glucose coming together with another glucose, that gives you maltose. So know that two glucose coming together to form a disaccharide gives you maltose. Also know that when glucose comes together with fructose, that you get sucrose. And sucrose is just simple table sugar. Also, disaccharides have a formula of CnH2n minus 2, O to the n minus 1. So let's just pick a random number. Let's do 3. So if our n is going to equal 3, then it would be C3, H, 4, O, 2. All right, so polysaccharides are just polymers of sugars. So they're just many sugars, and these also come together through dehydration. So polysaccharides are used for storage, and they play very important structural roles, and they have a formula that you can see right here. Again, you're going to want to know this as well. Make sure that you can tell the different formulas between the monosaccharides, polysaccharides, and disaccharides apart. So we have different types of polysaccharides. The first is starch, which is just a huge chain of sugar made by plants. We do not, animals and humans, we don't actually make starch, but we will eat them from the plants. And a good example of this is a potato. You know, potato is a starchy vegetable. We also have glycogen, and glycogen is just the storage form of glucose in animals, and this will get stored in your liver and in your muscles. You also have cellulose, and cellulose is what makes up the plant cell wall, and we'll talk more about that later on in our cell lecture. Cellulose is also not made by humans, and we can't digest it either. In fact, not many things can. Cellulose, when I think about cellulose, I think about cows eating grass in a pasture. The grass is full of cellulose, and cows are one of the only things that are actually out there that can digest the cellulose. But when you think of cellulose, you can also think about fiber. So whenever your mom or a doctor has told you need to introduce more fiber into your diet, well, that's that cellulose in those cell walls. So that fiber will actually help your intestines to digest food and to eliminate waste products. And then we have chitin, and chitin is found in the shells of insects and crustaceans like lobster or crab. It's what gives them that really hard outer shell. So polysaccharide starch, again, we don't make this as humans or animals. These are only made by plants, and the plants actually make this in the chloroplast, and this is where photosynthesis has occurred. And I'm sure you guys have heard of that before. If not, we're going to talk about it more later on. But through the process of photosynthesis, the plants make starch, which is just their storage form of sugar, which then we eat. So starch is made by plants, and you see an animal or a plant cell here. We have a chloroplast, which is where photosynthesis occurs, and that's how starch is made. And starch is just how it's the storage form of sugar for plants. Now remember, only plants can do this, not animals, because we don't have chloroplast to do so. However, we do take the starch from plants and use this as energy. Glycogen, it's the major storage form of glucose in animals, and glycogen is stored in the liver. Now, you do want it in a certain moderation. However, if you eat too many carbs, you can have too much glycogen stored in your liver, and that can actually cause a fatty liver, which is not a good predicament to find yourself in. Cellulose is just a polymer of glucose. Remember that this is only made by plants. Cellulose is not digestible by animals, or not by humans anyway. Cows are able to break it down, and then termites, they themselves cannot break it down, but they have a certain enzyme in their gut that allows them to process it. All right, so there's not a very big difference between starch and cellulose whenever it comes to orientation. They're actually very similar. You can see here that the OH group on starch, they're always downward, as in cellulose, they go up and down on every other molecule. So cellulose is a major component of the plant cell wall, and in fact, plants produce hundreds of billions of cellulose per year. And then we have chitin. Again, know that chitin is what's in that outer shell in insects and crustaceans. It's also used to make surgical thread, and then it's also used again as a type of fiber when it comes to digestion. So if you were to look on the back of a nutrition label and you saw that your food consisted or had chitin in it, it's probably some ground up shrimp shell or some kind of crustacean. It's not going to be one of the bugs. All right, so next is lipids, otherwise just known as fats. Now, lipids are hydrophobic, and you should remember that hydro means water, phobic means scared of. So they are hydrophobic, meaning that they don't like to be around water. So they're insoluble in water. Now, if you remember that during our water lecture, we talked about how water is polar and how fats or lipids are nonpolar. So they will repel against each other. So lipids play very big functions, or have very important functions when it comes to us as animals. They play a big role in energy storage. They help cushion vital organs. They also play a role in insulation. So if you have a friend who's really skinny, or if you're losing a lot of weight, you're going to have a harder time holding in heat versus someone else who has more fat. Okay, so fats are made from triacylglycerol, also called triglycerides, and they are made of one glycerol, which is just a 3-carbon alcohol, and they usually have 3 fatty acid tails. However, this is not always the case. So you can see here our glycerol with its 3 carbons, and then we have a fatty acid tail right here. So triacylglycerols or triglycerides are formed through a dehydration reaction, and if you remember, that's where we're losing a water molecule. And whenever this occurs, we are left with an ester linkage holding our fatty acids to this glycerol backbone. Fats are made of carbon, hydrogen, and oxygen, but unlike our saccharides, they have way more of these. So if you were to see a molecule with like 153, you would know that that's a fat rather than a saccharide. Okay, so we have a couple of different types of fatty acids. The first is saturated, we have unsaturated, and we also have phospholipids. So saturated fatty acids have no carbon-to-carbon double bonds. I don't want you to be confused here by this double bond between this oxygen and carbon. We don't see any double bonds between any of the carbons, so we know that this is going to be a saturated fatty acid. And because we don't have any double bonds between these carbons, that gives us more room to add more hydrogens. So it's saturated with hydrogen, which is what makes it a saturated fatty acid. So a couple examples would be butter, lard, ghee. These are all things that are solid at room temperature, and you want to limit your consumption of saturated fatty acids since they are solid at room temperature. If you eat too much and it gets in your blood, that becomes solid. It becomes an issue whenever it comes to blood circulation. So saturated fatty acids have one or more double bond between our carbons. We see this one right here. So these are not solid at room temperature, and that's because this little kink right here makes it more difficult for these molecules to lay on top of each other so they don't become solid. A couple examples would be olive oil, sunflower oil, peanut oil. So then we have our phospholipids, and phospholipids have two fatty acid tails, which we see here, and they're combined to a glycerol backbone. So we have our hydrophilic head, and you should remember hydro again means water, philic means like. So our glycerol, it also has a phosphate attached to it, where that third fatty acid would normally be. We have a phosphate group instead. So you see that phosphate group, that kind of gives you a hint that you are dealing with a phospholipid. And then we have the glycerol, and then we have two fatty acid tails. So again, so this head right here is water liking. And then we have our two hydrophobic fatty acid tails. So if this were to go into a water solution, our hydrophilic heads would automatically line up towards the water molecules, and our hydrophobic tails would want to go inward. So we see that here in our phospholipid bilayer, and this is, we're going to talk about this later on in our cell lecture, but we can see our hydrophilic glycerol phosphate heads facing towards the water on both of these sides, and then our hydrophobic fatty acid tails congregate together inside. Okay, so let's do a quick recap. We should now have a better understanding of the synthesis of macromolecules. Remember, to form bonds, we use dehydration reactions, and to break bonds, we use hydrolysis. We have talked about monosaccharides, disaccharides, and polysaccharides. We've talked about the four major types of lipids, and then we've differentiated between saturated and unsaturated fatty acids. All right, guys, I hope this was helpful. I'll see